So yeah, we're going to be presenting our ISP project, which was on midbrain reticular nucleus's control over pupil dynamics and how this correlation is modulated by the visual contrast presented in the organism. And our team comprises of me and my mentor, Dr. Bianca Sieveritz. Let's get to the presentation. It's not going to the next slide. Okay. So what is reticular formation? Reticular formation is a complex network of brainstem nuclei, and it has several functions. Like it's involved in motor control, it's involved in some other emotional control. And we are particularly interested in the role of reticular formation in pupil dynamics. And this is a section, this is an image, representative image of a mouse, a rat brain. And this you can see the reticular formation here. And there are several regions in reticular formation that are involved in pupil dynamics, one being medullary pontine reticular formation, which is involved in gaze holding and also head movements. And then there is paramedian pontine reticular formation, which is involved in horizontal eye movements. And then there is midbrain reticular formation, as this is an image from Alan Moss print atlas. And this purple region is the midbrain reticular formation. And this particular region is known to be involved in vertical and horizontal eye movements. And our hypothesis was to further explore and how mRNA activity is correlating with the mRNA's midbrain reticular nucleus. So we wanted to find out how exactly is the correlation looking like and what is the magnitude of this correlation based on the visual stimulus presented to the organism. And for this, we used the open source data set by Sieveritz, and this is the paper name. And what the experiment done by Sieveritz is like this. So mice are head fixed and they're presented with 3 screens in front of them. And then the brain is also, the neuropixel probe is also implanted into different brain regions in the mice. And when they perform the task, the neural recording is taken. And what is presented on these screens? So it looks like this. So visual contrast discs are presented on the right and left side of the screen. So this is the left screen, this the right screen, and then mice is provided with a disc. So what the mice has to do is to move the higher contrast disc to the center of the screen, to the center screen. If the high contrast is on the left side, then the mice has to move that screen towards the center. While if the high contrast is on the right side, the mice has to move that contrast to the center. So that was a task given to the mouse. And there were different combinations of contrast on left and right side that the team presented the mice with. So it looked like this. So there's a contrast level of 0, 25% contrast between the lines in the disc, 50% contrast between the lines in the disc, and 100% contrast. And same for the left side and the right side. So overall, there are this many combinations of contrast tasks that were presented in front of the mice. And the mice viewed the stimuli for like 500 to 1,200 milliseconds before they made the decision to turn the wheel to left or right. And so since this 500 millisecond window is constant for all the mice, so we selected the first 500 millisecond of the stimulus presentation window for our analysis. So in the Sieveritz data set, they have the data from a wide range of brain regions, as you can see in the image, but we were particularly focusing, as you already know by now, on midbrain reticular formation. So all the neural recordings were from the left hemisphere of the mice brain, and we had a total of 1,060 neurons, mRNA neurons recorded from 7 mice over 11 sessions. It was multiple trials. Several combinations of left and right contrasts were presented in front of these different mice. And also, Sieveritz dataset also had eye data, that is horizontal eye movement data of the mice, vertical eye movement data of the mice, and also pupil area data of the mice. And all these were recorded from the right eye. And So what we try to do is to find the cross correlation between the MR and spike trains recorded from the midbrain reticular nucleus and the horizontal eye movements, and then cross correlation between the MR and brain activity and vertical eye movement, and the cross correlation between MR and activity and the pupil area. And we used a lag of 10 millisecond for our cross correlation. So we shifted MRN versus horizontal eye movement by a 10 millisecond lag each time and recorded the cross correlation. And then after that, we get something like this. So for one neuron, for one particular condition means one particular combination of left and right stimuli. This is how the graph looks after we calculate the cross correlation across different time shifts. And after this, what we did is we picked out the maximum cross correlation and the minimum cross correlation for each neuron for each condition, and then we averaged it. So finally, we got a mean maximum cross correlation for each condition and a mean minimum cross correlation for mean minimum correlation for each condition. By condition, again, I mean the different visual stimulus that was provided in front of that was presented to the mice. And now let's come to our final data visualizations. So when we plotted the mean maximum cross correlation and pupil area, we plotted it across a constant contrast level on left and right side. That means we selectively picked up visual contrast levels situations which monotonically increased on both sides. That is 0, 0 on both sides, or 25%, 25% on both sides, or 50%, 50% contrast on both sides, or 100%, 100% contrast on both sides. So equal contrast levels we picked up such condition, and that's the x-axis. And then on y-axis, we correlated the mean maximum cross correlation. And for pupil area, what we could observe is that when the contrast level is beyond 0, that is when it increases, we see a rise in the maximum cross correlation. So mean maximum cross correlation becomes larger when the visual stimulus contrast is larger than 0. Same thing when we plotted for pupil horizontal data, horizontal eye movement data, which is these red dots. We could see a similar trend. That is when the visual contrast level is beyond 0. Again, the maximum cross correlation is significantly increased. And same thing again for pupil vertical, that is vertical pupil movement, that is these yellow triangles. And we could see that again, a very similar trend of when the contrast levels are more than 0 on both sides. We could see that the mean maximum cross correlation has significantly increased. So now we did the same thing for minimum cross correlation, and we found the exact opposite trend. That is, for pupil area first, when the contrast level is more than 0, we could see that the minimum cross correlation is dropping down for all the higher values of contrast levels. Same for pupil horizontal, that is the minimum cross correlation value is dropping down when the contrast level is beyond 0. And same for pupil vertical, which is pupil horizontal here is the red one, and the pupil vertical again, the yellow triangles, that when the contrast levels are beyond 0, we could see there's a significant drop in cross correlation, minimum cross correlation for all the three metrics, all the three pupil metrics. So this is the graph. This is the figure of both the graphs together for mean maximum cross correlation and minimum cross correlation. And we can again see that when the correlation between neural activity and subsequent pupil dynamics. For all the three dynamics we looked at, pupil area, pupil horizontal, pupil vertical, all of them, the correlation between these two dynamics and neural activity becomes stronger when the visual contrast stimulus is larger than 0 on both sides. And we also did another sort of visualization. We grouped the stimulus into low and high. That is, aggregated means across. Low means when both the sides are 0, 0, or both the sides are 25, 25 contrast levels. And high means when both the sides are like 50, 50 contrast levels or 100% contrast levels. So that is the low. And we plotted the left side stimulus strength after grouping them into low and high on the x-axis. And then we plotted again the mean maximum cross correlation on the y-axis. And the plots, the red color shows the high values for the right side. That is, here, it means the left side stimulus is high, and the red one, the right side stimulus is also high. And blue means left side stimulus is low, high, but the right side stimulus is low. That is, it could be either 0 or 25%. So that's how the plot looks like. That's how the plot was plotted. And what we could see again was, for example, in the low situation there is not much of a difference. But when it comes to the high left side stimulus strength, we could clearly see that the highest correlation value was when the right side was also high. That is, when the right side and both left side stimulus strength, contrast strengths were high, the correlation between the brain activity and the pupil area was the highest. Similarly, the circle is now the pupil horizontal, and across was the pupil area. Again, when the left side stimulus is high and the right side stimulus is also high, the correlation is higher than that when the right side stimulus is low. Again, for pupil, we plotted all the three pupil metrics: pupil vertical, pupil horizontal, and pupil area. We could again see the same trend. Similarly, we did it for minimum cross correlation as well. And here also, we could see that when for example, when the left side stimulus is high and the right side stimulus is also high, that is when the minimum correlation is the least. And when the right side stimulus is high and the left side stimulus, yeah, that's how the graph. And similarly, we could see that as we could see a very similar trend for pupil horizontal, that pupil horizontal, where when both the contrast levels are high, the mean minimum cross correlation is the lowest for pupil horizontal. And same, when all the three pupil metrics were plotted together, we could see that when both the stimulus, both the side contrast levels were high, it showed least minimum correlation at maximum positive correlation. So that means our results indicate that when pupil activity is clearly correlated with pupil size, horizontal gaze, and vertical gaze, and the correlation magnitudes between the brain activity and each of these pupil metrics are almost similar in magnitude, as you can see from the graphs. And also, the correlation strength depends on the visual contrast of the presented stimuli. As we can see, when the contrast level presented to the mice was high, then the correlation is also high. And our results so all the literature I could find mostly talked about how MRN activity is related to gaze control, that is horizontal eye movements or vertical eye movements, but I didn't find any which specifically talked about how MRN is related to pupil size metric. But through our exploration, we could figure out that similar to how MRN is correlated with pupil horizontal and pupil vertical gaze, it's also correlated with pupil size. So that's something new that we found. And all the three correlations are of similar trends, showed similar trends. And also, MRN's involvement in pupil and gaze control both are similarly influenced by the visual contrast strength presented in front of the mice. That's all we did for a small presentation from this small group. I would like to extend my very, very warm gratitude to my mentor, Dr. Bianca Sieveritz, for guiding me and helping me a lot. And also Neuromatch Academy and Impact Scholars Program for giving me this opportunity. And I also thank my former team member, Karishma, who helped me with the initial data exploration. And yeah, thank you all for listening.